Human Embryology. Lecture Plan. Embryology. Theme: the structure of germ cells, stages and process of embryogenesis, fertilization, cleavage, gastrulation, neurulation, histogenesis, extraembryonic organs. Embryology is a science about the embryo origin and development. Contemporary biologic sciences recognize general and special embryology. The first one studied the most common features of embryo origin and development. Special embryology, science about the individual development of particular group of animals, classes, and so on. The embryogenesis means the development of the embryo. The structure of germ cells. Germ cells means spermatozoa and oocyte. Spermatozoa is the male reproductive cells. They are 60-70 micrometers long and have a very specific shape. Each spermatozoan head has a head, neck, and tail. The head of spermatozoa is of oval, flattened shape. It is the most suitable shape for passage forward due to the moving of flagella. There are 23 chromosomes. 22 chromosomes is autosomes, and last one, chromosomes X or Y chromosomes. Most of the sperm cytoplasm is eliminated during maturation, leaving only certain organelles that are modified for spermatic function. There is a special structure, acrosome, or acrosomal vesicule. At the top of spermatozoan head, it is a specifically changed Golgi apparatus which contains enzymes hyaluronidase and trypsins. There is a special structure, acrosome, or acrosomal vesicule, at the top of spermatozoan head. It is a specifically changed Golgi apparatus which contains enzymes hyaluronidase and trypsin. The neck contains a proximal centriole. The tail consists of 3 parts: intermediate parts, main parts, and terminal or ending parts, terminal parts. The main component of all tail are axial fiber. The intermediate part also contain many mitochondria. Mitochondria have very long and spiral shape, which surrounded axial fiber. The main part contain axial fiber and contain a special proteins which surrounded axial fiber. The end part contain only axial fiber. Each spermatozoon is able to travel long distance by moving its tail. The tail is a flagellum. Flagellum, it is a complex structure. The major motor portion of the flagellum is called the axoneme or axial fiber. We also called axoneme. It is formed by macrotubules, which consist from centriole, which start from centriole at the base of the sperm nucleus. The core of the axoneme consists of 2 central microtubules surrounded by a circle of 9 doublet microtubules. Actually, one of the microtubules of each doublet is complete, having 30 protofilaments. The other is C-shaped and has only 11 protofilaments. Although tubulin is the basis of the structure of flagellum, other proteins are also critical for flagellar function. The force of for the spermatozoa propulsion is provided by dynein, a protein that is attached to the microtubules. Dynein hydrolyzes molecules of ATP and can convert the released chemical energy into the mechanical energy that propels of the spermatozoa. Oocyte or ovum is female germ cells. It is the largest cells in human body, about 130–150 micrometers in diameter. In a human body, and these cells cannot move. It is covered with a few tunics. From outside, they are covered by corona radiata. First tunic, corona radiata, or tunica granulosa. Then tunica zona pellucida. Second tunic is zona pellucida. And third tunic is avalemma. The avalemma is the cytolemma. Zona pellucida, this is a special chemical membrane which contain much glycoproteins and glycosaminoglycans. The most important glycoproteins are ZP1, ZP2, and ZP3. They take part in a process of fertilization. ZP3 is a receptor for spermatozoa. ZP2 and ZP3 also prevent polyspermia. The outer part, the corona radiata, consists of numerous follicular cells, which give their process to the ovum through the previous tunic process. Their function are protection and nutrition of the oocyte and regulation of its maturation. Nucleum of the oval, located and contains 23 chromosomes, 22 autosomes, and last one, only X chromosomes. There are all the organelles of a general meaning in the oocyte cytoplasm. Apparatus Golgi is especially well developed and produces yolk inclusions. In the peripheral layer of the cytoplasm just under the plasmalemma, numerous cortical granules may be seen. They take an active part in the process of fertilization in cortical reaction. This microphoto you can see oocyte without corona radiata, and this oocyte contained also zona pellucida, transparent layer, white transparent layer, and very thin ovolemma, cytolemma, very thin. This is cytoplasm of oocyte, and the cytoplasm you can see that cytoplasm contains a lot of granules. This pronucleus. This is pronucleus. The next stage process of embryogenesis. The first stage in embryogenesis is fertilization. Fertilization is the first stage of embryogenesis, the process by which the male and female gametes fuse and result in a zygote formation. There are two stages of fertilization: distant stage and contact stage. Distant stage of fertilization include two process of capacitation and process of taxis. Capacitation means special activation of spermatozoa, which occurs with them in female reproductive organs. Only after such changes they began to move forward. Taxis is the active passage of spermatozoa. There are hemataxis and there are rheotaxis. Hemataxis is our hematropism. This is moving spermatozoa toward to the oocyte because oocyte produce special attracting chemical substances, gammons. Rheotaxis is moving against the fluid flow in the uterine tube, moving of spermatozoa against the fluid flow. The next stage of fertilization is contact stage. Contact stage of fertilization include three processes. Process of penetrate the corona radiata, penetrate the zona pellucida, and penetrate the avellana, and then syngamy. Contact stage. In female reproductive organs, this process takes place in ampullar portion of the uterine tube. This is process of contact spermatozoa and oocyte. Contact stage have three processes: penetrate of the corona radiata, penetrate of the zona pellucida, and penetrate the avellana or syngamy. Penetration of the corona radiata. About 200–500 spermatozoa surrounded the egg or oocyte and join into corona radiata. Due to the fact that spermatozoa tails move, the oocyte begins to rotate too, and the corona radiata cells, follicular cells, was dispersed. Then, next stage, penetrate the zona pellucida. Only one spermatozoa can penetrate of the zona pellucida. When this spermatozoa contacts with ZP3 glycoproteins of zona pellucida, acrosomal reaction begins. What does it mean, acrosomal reaction? Acrosomal reaction, this is a release of acrosomal enzymes from acrosome. Acrosome, this is special vesicles which are located in upper part of spermatozoa head. And acrosomal enzymes is trypsin and hyaluronidase, which can destroy zona pellucida, proteins of zona pellucida. This reaction allows spermatozoa to penetrate the zona pellucida and then to come in contact with the plasma membrane of oocyte. In these diagrams, you can see that spermatozoa moving closely to oocyte and contacts with zona pellucida, and acrosomal reaction begins at that time. Acrosomal reaction allows to destroy part of zona pellucida, and spermatozoa can penetrate this zone and to make contacts with cytoplasm of oocyte. Cytoplasm of spermatozoa fuse with cytoplasm of spermatozoa fuse with cytoplasm of oocyte, and their cytoplasm fuse also. Then, penetration of avellana. After adhesion, the plasma membranes of the spermatozoon and the oocyte merge, fuse, then spermatozoon nucleus, mitochondria, the part of acronym enter in the ooplasm, but the spermatozoon plasma membrane is left behind on the oocyte surface. As soon as the spermatozoon has entered to the oocyte, the oocyte responds by two ways: cortical reaction and resumption of the second meiotic division. Cortical reaction. So after penetrate of our limb, we can see cortical reaction. Cortical reaction. This is release of cortical granules from cytoplasm of oocyte to space between limb and zona pellucida. Cortical granules are released by exocytosis. As a result of the release of cortical oocyte granules, which contain lysosomal enzymes, the oocyte membrane becomes impenetrable to other spermatozoa, and the zona pellucida change its structure and composition for sperm binding and prevent penetration. So, zona pellucida become more hard. This reaction prevent polyspermia. What does it mean, polyspermia? Polyspermia is penetration of more than one spermatozoon into the oocyte. The zona pellucida becomes a fertilization tunica. The space between the avalemma and tunicle of fertilization is perivitelline space in which water is stored. Resumption of second meiotic division, second process. Resumption of finishing of second meiotic division. The oocyte finishes its second meiotic division immediately after entering the spermatozoon. One of the daughter cells, which receives hardly any cytoplasm, is known as a second polar body. The other daughter cells, it is definitive oocyte, and its chromosomes 21, 22, plus X chromosomes arrange themselves in the vesicular nucleus known as a female pronucleus. Then, after, we can see syngamy, the last process in fertilization, syngamy, the process of nuclear fusion. The spermatozoon nucleus, male pronucleus, and female pronucleus. At the moment of their fusion, such unicellular organism is named synkaryon. As the result, a zygote is formed, which consists of 46 chromosomes. The next process after fertilization is cleavage. Cleavage, or fissure, special mitotic division of zygote without growth of daughter cells. Human zygote has a full subequal asynchronic type of cleavage. Human zygote has full subequal asynchronic type of cleavage. It means all the zygote is dividing into blastomeres, full. And all the blastomeres are not almost equal in size. One of blastomeres is a little bit smaller and lighter than the other one. Different blastomeres are dividing at different time asynchronically. Their amount changes such way: 2, 3, then 5. At the third during second, third, or fourth day, blastula consists of several blastomeres, about 16 blastomeres, which are tightly connected one to each. This stage have a name, morula. This is morula. At the 5th or 6th day, embryo becomes a next stage, blastula. Become a blastula, which consists of a lot of blastomeres, about 100. During all this period of time, blastula has a constant size and is surrounded with a tunica of fertilization. Tunica of fertilization is former zona pellucida of oocyte. At that time, blastula consists of larger, dark blastomeres, embryoblasts, which are surrounded with the smaller light cells, blastomeres. trophoblast and contain the space inside blastocell. Such type of blastula is called blastocyst. So in these diagrams you can see stage of two cell stage, only two blastomeres you can see, and you can see that one blastomere is more large than other. Then about 20-30 hours we can see four cell stage blastomeres, and blastomeres have a different size. In this diagram you can see 8 cell stage blastomeres. In the stage you can see morula. Morula contain about 16 blastomeres, which some of these blastomeres have more large size and they're more dark color. Some of these blastomeres more light color, more less size. And in this diagram you can see that all this stage, all this embryo surrounded tunica of fertilization. Tunica of fertilization, this is forming zona pellucida of our site, but after cortical reaction, zona pellucida become more hard, more impenetrable for other cells, for other maybe microorganisms. Then, size of embryo cannot change because zona pellucida cannot change their size. Next stage is blastula. Blastula contain a cavity inside. Morula don't contain a cavity inside because all cells have closely contact with each other. In blastula, we have cavity with a tissue fluid, blastocell, and we can see inner group of cells, inner cell mass, more dark and more large cells. This is future embryoblast. From these cells, future embryo will develop. Outer cells is trophoblast. These cells more light and they have a less size. It is future extraembryonic organs. And then in this stage, early blastocysts, you can see they have a tunicle fertilization. This is light blastocyst without tunicle fertilization. And this blastocyst after when they lose their tunicle fertilization, start implantation, the next process. Embryo hatching from the zona pellucida, later stage of blastocysts, when blastocysts leave tunicle fertilization and going to implantation in the wall of uterine. Then the next stage is implantation. After cleavage, we can see implantation. What does it mean? Implantation, this is process of dipping embryo inside wall of the uterine. By the end of the first week, 7 days of the development, the blastocyst has begun its implantation into the uterine mucosa. Implantation is the process of blastocyst introducing into the endometrium. There are two stages of implantation: adhesion to the surface of endometrium and invasion, dipping into the endometrium. There are two stages of implantation: adhesion to the surface. There are two stages of implantation: first stage adhesion and second stage invasion. Adhesion. This is contact of blastocyst with the surface of endometrium. in this diagram, contact of blastocyst with the surface of epithelial surface of endometrium. Second stage, invasion, dipping of blastocyst inside into the endometrium, like in this diagram. Blastocyst dipping inside connective tissue of tunica mucosa of endometrium. Process of implantation lasts all at about 40 hours till the end of second week. The embryo is completely embedded in the endometrial stroma. Stroma does it mean connective tissue, and covered with epithelium above. The trophoblasts penetrate continuously deeper into the endometrium, thereby differentiating into the syncytial trophoblasts and the cytotrophoblasts. In this diagram, you can see dividing of trophoblast. This part of trophoblast, blue color, become cytotrophoblast. This part of trophoblast become syncytial trophoblast. Syncytial trophoblast produce special enzymes which can destroy maternal tissues, connective and epithelial tissues of a uterine wall, and then destroy wall of capillaries, and it need syncytial trophoblast need for to make contacts with maternal tissues or tissues of a uterine wall. During the first 30 hours after fertilization, it possesses nutritive substances from the yolk inclusions of oocyte. It is so-called vitellotrophic nutrition. After this vitellotrophic nutrition, histiotropic nutrition begins. This during the first month of pregnancy, embryo is fed with the secretion of surrounded tissues, feeding by substances in surrounding connective tissues. At the period of embryogenesis, hormone progesterone is necessary for very much because of its influence of the secretory process. Then, at the beginning of the 5th week, trophoblast enzymes dissolve the wall of uterine mucosa vessels and new type of nutrition begins. It is a hematotrophic nutrition because the embryo receives all the necessary products, nutrition from the mother's blood, from these vessels with mother's blood. The next process is gastrulation. Gastrulation is a process of bilaminar and three-laminar gem disc formation. It is the next stage of embryogenesis after cleavage and after implantation. It occurs in the embryo in the course of implantation and lasts from 7th till 17th day. There are two stages of gastrulation: early stage of gastrulation and later stage of gastrulation. Early stage of gastrulation starts from 7 days. Later stage of gastrulation starts from 14 days. As a result of first ones, two gem layers appear, ectoderm and endoderm. The third layer, mesoderm, is developed during the later gastrulation. The early stage of gastrulation, these diagrams. This is morula, this is early blastocyst, and this late blastocyst, and which become gastrula in process in early stage of gastrulation. The early stage of gastrulation occurs through delamination. This process have a name, delamination. It means tangential splitting of blastomeres into two layers. Blastomeres forms two layers, epiblasts and hypoblast. In this side, we can see epiblasts, or in another name, primary ectoderm. In this side, we can see hypoblast, or another name, primary endoderm. The epiblast cells form a cavity known as amniotic sac. Epiblast cells divide and forms cavity, amniotic sac. The hypoblast cells divide and forms yolk sac. In this part, we can see yolk sac. The place where of both sac contacts has a discrete shape, thus the proper gem disc. This place where both sac can forms contacts have a name, gem disc. From this part produce embryo tissues. Other parts, these and these, produce extra embryonic organs. For example, these cells produce yolk sac. Yolk sac need for nutrition. And this part produce amniotic sac. Amniotic sac can produce amniotic fluid for liquid environment for development of embryo. Then later stage results in three laminar embryo disc formation through migration. So we have process migration in this stage. What does it mean, migration? It means moving of different cells in different directions. The cells of a gem disc, or rather cells of epiblasts, and migrate from cranial to caudal part of an embryo and then return back together, making the primitive streak and primitive node. In these diagrams, please. So, gem disc, amniotic sac, yolk sac, and the place where they when they contact this place have a name, gem disc. This is future embryo. This is future amniotic cavity. This is future yolk sac with the nutritive substances for nutrition. Gem disc, epiblast in gem disc. In a later stage of gastrulation, cells of epiblast migrate in different directions. As a result of this moving of cells, primitive streak is formed. It's like this primitive streak. A little invaginations in cell is primitive streak. And also primitive node. This part have a name, primitive node. Little invaginations in epiblast. Then cells of primitive node migrate in place between epiblast and hypoblast, and they form notochord. The cells of primitive streak migrate in space between epiblast and hypoblast, forming a third layer, mesoderm, like this, mesoderm. Thereby a three-layer, trilaminar embryo forming. It has an ectoderm formed epiblast, or primary ectoderm, mesoderm, and euler, and entoderm or primitive primary entoderm or hypoblast. So we have three laminar embryo. Next stage is histogenesis, and histogenesis is forming of different organs and system of organs of embryo and tissues from ectodermal layer, entodermal layer, and mesodermal layer germ layers.